Hello students, today I'm going to start a new topic. Okay, last time we discussed on restriction enzyme, right? So today in chapter Biotechnology, I'm going to start Gel Electrophoresis. Now before understanding this process, let's go with the basic concept, okay? For gel electrophoresis, you need a target DNA, or I will say that isolation of DNA. Isolation of DNA. When I say isolation, it means that extraction of DNA. Now, it depends on the experiment that from where the DNA will be isolated. Some can isolate from the plants, we can also isolate from the animals, we can also isolate from the fungi, okay? Likewise, after isolation of the DNA, we have to just say that this is our DNA. As I said that for isolation, you need enzymes, right? Last class I have already told that we need enzymes to break the cell wall, okay? For example, like chitinase, in the case of fungi, right? Like lysozyme, for the case of bacteria, right? Cellulase. So this enzyme is for plants, okay? Now if you want to isolate DNA, first of all you have to break the cell wall, okay? After that, there may be some proteins. For example, in the case of eukaryotes, as I said, that histones are present, right? So there may be some histones, or there may be some RNA, okay? Now if there is a histone, then remove with the help of, it's a protein, so use protease, digestive enzyme. So with the help of protease, you can degrade the protein molecules. And if there is a RNA, you can go for RNase. Okay. So likewise, or you can say that ribonuclease. Okay. Right. So this can be removed. This can be degraded. Then you get only your, in a test tube, you can get only your DNA fragments. Okay. Now, there may be histone, protein, there may be RNA, but all these will be degraded with the help of enzymes. After degradation, we have to use you have to add chill ethanol. Okay, this was out in your medical examination in 2017. Okay, so we have to add chill ethanol, and then it will be precipitated out. Once precipitated out, then you get your DNA. And this process is called as spooling. Okay? Now, this is the method for the isolation of DNA. For gel electrophoresis, you need a DNA fragments. Okay? Once that DNA fragments or DNA has been extracted, now, so this is the DNA that you have extracted, and now we have to use restriction enzymes or restriction endonuclease to get fragments. So these restriction enzymes or endonuclease will make the DNA fragments. So for gel electrophoresis, we need DNA fragments. Okay. So next is said to be agarose. So what is agarose? Agarose is a natural polymer. Okay. Natural polymer which was extracted from seaweeds. Okay. Extracted from seaweeds. Now this is in a solid form, okay, like a powder, okay, in a powder form. Now this you have to take in a conical flux, okay, just say that this is your conical flux, 250 ml, and then you have to add agarose. Once you add agarose, you have to add some water or some buffers, okay, and then we will keep in an oven for some times, and then this solid will get in a liquid form, clear distilled liquid form, and then we will put in a tray. Just say that this is a tray, and then you're going to put this agarose here. Actually, this is in a liquid form, so we will let it solidify. Okay, we will let it solidify, keep it for some time, it will get solidified, and it is just like a jelly material, okay. So agarose is a matrix, okay, it's a matrix where we are going to insert our DNA fragments. So the first is that you need a DNA fragments, and how to do that? Isolate the DNA, after that cut with the cleave with the help of restriction enzymes. Then after that, what we need? We need agarose, which is a matrix, okay, where we are going to insert our DNA fragments. The third is said to be buffer. Buffers are in a liquid form, and we have to prepare that. Example like TAE, okay, tris acetate EDTA. It's a common buffer that we use, and then once you prepare the buffer, actually the functions of buffer is to maintain pH. Maintain pH. As I said in the molecular basis of inheritance, that DNA is a negative charge, right? So when I say gel electrophoresis, it is a machine, okay? Actually it's a machine which runs with the help of electric currents. So just say that this is your gel electrophoresis machine. So there will be cathode and there will be anode. Cathodes are usually positive charged or negative charged, okay, and anodes are positive charged. So this is a machine, it has a wire connected at the circuit, okay, current will pass, okay. Now here there will be two blocks. In the first block, okay, we are going to put our agarose. Just say that this is a agarose, this is a matrix. Now we are going to put a comb. You know, this is a comb like, okay, we put a comb for some time, and then if you take out, a wells will be developed like this. Okay, we'll put a comb for some time, and then if you take out, then there will be wells like that. This is our agarose. Now here we put a buffer. Once we add buffer, once we set up the agarose, now we are going to put, just say that these are DNA fragments which was generated with the help of restriction enzyme. Okay, here we are going to put now ETBR. This is a dye that is going to give color to our DNA fragments. So ETBR is nothing but ethidium bromide. It's a radioactive substance. It's a carcinogenic substances. Okay, so it has to be handled with care. Now once it is added, we have to mix it thoroughly, and then we have to insert here with the help of micro pipette. Okay, just say that you have inserted your DNA fragments here. Remember that the DNA fragments have to be put, since it is a negative charge, we have to put at the cathode. And once an electric current is passed, what will happen is that these DNA fragments, some are smaller in size and some are large fragments. So gel electrophoresis are used to separate the DNA fragments of different sizes. So when it is run in this machine, in the gel electrophoresis, just say that these are large fragments. can see that these are large fragments and these are small fragments. So the negative charge will move towards the positive charge, right? So since DNA is negative charge, it moves toward the anode side, okay? And you can see that the smaller fragments you can see the smaller fragments and you can see the large fragments. Large fragments. Okay? Now these large fragments will be near to the well. Okay? And the smaller fragments will be away from the well. Okay? Now, how will I know that this is, just say that this is our target DNA? So, these are designed, these are identified with the help of molecular markers. I'll just write here, molecular markers. These molecular markers will identify the size. Just say that this is 5 kb kilobases. Just say this is 3 kb kilobases. Just say that this is 1 kb kilobases. So if our target DNA, if we know our target DNA, okay, if our target DNA is 5 kilobases, then this molecular marker will locate, okay. When we perform this experiment, the molecular markers are attached at the site, okay, and when we see that in this line, if this is 5 kb area, then this is our target DNA, okay. So in gel electrophoresis, we separate the DNA fragments, okay, based on the size. Different size of DNA fragments can be separated for the use of rDNA technology. Now once the process is over, then we have to take out this gel and then we have to put in a UV light. Observe under UV light. If you see in UV light, if you observe in UV light, you will see orange colored band. Orange colored band will be observed. Now once you know that this is your target DNA, then you have to cut this gel and you have to extract out. So see, just say that this is your target DNA with a gel, okay. Now this process, the extracting, okay, of your target DNA or your DNA fragments from the gel is called elution. Okay, so this process or this technique is very important for the separation of the DNA fragments. Now in the next class, we're going to talk about the cloning vector, okay, the vehicle that will carry the DNA fragments. Which are the vehicles that can be used in rDNA technology? Thank you.